All right, let's dive into a piece of technology that's kind of like a superpower. It lets us see the chemical secrets hidden inside of everyday things. It's called diffused reflectance spectroscopy, and it's this amazing way to understand the world around us without ever having to break it open. Let's see how it works. So let's start with a puzzle. Imagine you're a food scientist and you need to know the protein content of some powdered milk. Or maybe you're an art historian looking at a priceless ancient statue and you want to know what pigments were used. How in the world do you analyze these things? They're opaque. You can't just shine a light through them. The standard technique, it's called transmission spectroscopy, does exactly what it sounds like. It measures the light that makes it all the way through a clear sample. And that's great for, say, a beaker of clear liquid. But for a solid pill, a piece of food, a painted wall, the light is just completely blocked. The traditional method, it just hits a wall, literally. And this is what we call the opaque object problem. Solving it meant we had to come up with a totally different way of thinking about how light and stuff interact. So, here's the big shift in thinking. Instead of trying to force light through an object, what if we just looked at the light that bounces off of it? That's the key. Transmission measures what passes through, but reflectance spectroscopy gets clever and analyzes the light that gets sent back at us. And this is where our story really kicks off. By seeing with reflected light, we open up a whole new universe of things we can analyze. But it's not quite as simple as just looking at a reflection, because it turns out there are two very different kinds of reflection. Okay, so when light hits a surface, some of it bounces off perfectly, like a mirror. That's called specular reflection. It's the shiny glare. It's clean, simple, and honestly, not very useful for figuring out what the object is made of. But, and this is the good part, another part of that light actually dives just below the surface, bounces around between the particles, and then escapes back out in all directions. That is diffuse reflection, and it is an absolute goldmine of chemical information. So this is the whole point. An entire field of science, diffuse reflectance spectroscopy, or DRS for short, was developed for one specific reason: to capture and make sense of this messy, scattered, but incredibly informative light. It's a technique that's designed to listen to the chemical whispers coming from inside the material itself. Now, catching this light is one thing, but how do we turn it into something we can actually read? How do we decode what it's telling us? Well, this is where we get to the idea of a spectral fingerprint. Think of it like this. Every single material out there, whether it's a pigment in a painting or a protein in a piece of food, it absorbs and reflects different colors or wavelengths of light in its own unique way. That pattern is its spectral signature. And just like a human fingerprint, you can use it to identify a substance with incredible accuracy. Right, but there's a catch. This diffuse light is, by its nature, messy. The signal we get is a jumbled mix of light that got absorbed and light that got scattered. So to get a clean, useful number, we need a way to mathematically untangle those two things. How do you do that? The answer, and this is really the secret sauce of the whole technique, is a brilliant mathematical model called the Kubilka-Munck theory. This is the key that lets us do real quantitative analysis. It gives us the exact formula to connect what we can easily measure, the reflectance, with what we really want to know, how much light the material is absorbing. Whoa, okay, don't let the equations scare you. You do not need to be a mathematician to get this. The important thing here is the purpose of this equation. It takes our raw measurement, which we call R, and it runs it through this function to get a new value, f of R. And here's the beautiful part. That new value is directly proportional to the concentration of whatever we're trying to measure. More stuff means a bigger number. It's as simple as that. And check this out. This shows you exactly what that transformation does. The bottom graph is the raw, kind of messy data we get from the machine. But after we apply the Kubilka-Munck magic, boom, you get the clean spectrum on top. All of a sudden, it looks like a perfect, easy-to-read absorption spectrum. This theory really does turn chaos into clarity. So we've got the theory down, but what does a machine that can actually perform this kind of magic look like? What's in the toolkit? Even though a full diagram might look complicated, a DRS system really just boils down to three main parts. You've got a light source, like a lamp. You've got a probe to shine that light on your sample and then collect the light that bounces back. And finally, you have a spectrometer, which is the brain that analyzes that reflected light and generates the spectral fingerprint. Now, to get really good, accurate data, you can't just point and shoot. There's a careful little calibration dance you have to do first. You gotta let the lamp warm up so its light is stable. Then you take a dark measurement with no light to zero out any electronic noise. After that, you measure a perfectly white reflective standard. And only then, when the instrument is perfectly dialed in, do you finally measure your actual sample. So where does a tool like this fit in? Well, you can think of it by comparing it to something simpler, like a colorimeter. A colorimeter is great at telling you how a human eye would see a color. It's like asking, is this red? But a spectrophotometer using DRS, that gives you the full story. It gives you the entire fingerprint, wavelength by wavelength. It tells you exactly what kind of red it is, down to the chemical level, which is why it's so critical for research and deep quality control. Okay, so we've covered the theory and the tools. Now for the really fun part. What is this actually used for? This is where it gets incredibly exciting, because the applications are just all over the place. Let's start with art conservation. Imagine being able to walk up to a 2,000-year-old Roman fresco with a handheld DRS device pointed at the wall and identify the exact chemical pigments the artist used. You can do that, all without taking a single physical sample. It's completely non-destructive, which is a total game changer for preserving our history. Or how about this? This quote from one of our sources is kind of mind-blowing. Think about the food industry. You can analyze the quality, the safety, the nutritional content of frozen food without ever thawing it out. That's a massive leap in efficiency and food safety. And the list just goes on and on. In pharmaceuticals, it makes sure every pill has the exact right amount of active ingredient. In medicine, scientists are using it to help doctors spot cancerous tissue without a biopsy. In agriculture, it can tell a farmer if their crops are healthy from far away. And yeah, even in space. It's how our rovers on Mars figure out what the rocks are made of. When you step back and look at it, from a priceless work of art to the medicine in your cabinet, DRS gives us this unbelievably powerful window into the chemical makeup of the solid, opaque world that we live in every single day. And that really leaves us with a pretty fascinating question to think about. If we now have the ability to read the chemical story written on almost any surface, what other incredible secrets are just sitting out there, hiding in plain sight, just waiting to be revealed?